This is a quick review on genetic mutations. We have chromosomal mutations. There's a couple different kinds. There's a deletion. You can see here the B section of this chromosome has been deleted. We have duplication, where the B section of the chromosome has been duplicated, so we have 2 copies of it. We can have an inversion, where we take the chromosome, it folds in on itself, and it cuts and sort of it reattaches, but in the wrong order. So instead of being A, B, C, then D, E, F, we have A, E, D, C, B, F. So the genetic information is not in the right order. And then we can also have what's called a translocation. When we have 2 non-homologous chromosomes, so 2 different chromosomes, you can see how the letters are different. They wind up exchanging genetic information with one another, and it gets all messed up. We also have what's called trisomy. Here's trisomy 18, where we have 3 copies instead of 2 of that particular chromosome. We can also have monosomy, where we have a loss of a chromosome. Oftentimes, these, if trisomies, monosomies will result in no individual being created, but sometimes they are, and then they have genetic disorders. And then we have point mutations. Point mutations are just what they say. They're a change in a single nucleotide. There are 3 types. We have a silent mutation, a nonsense mutation, and a missense mutation. Let's take a look at those here. If we have, here's our regular DNA without a mutation. Here's the amino acid that it codes for. If we have a silent mutation, we see the C became a T, but when we're looking at it on a amino acid level, the AAA and AAG code for the same amino acid. So really, this causes no problems whatsoever. In a nonsense mutation, we get a stop codon well before we are ready to. So what happens here is that the T got changed to an A, which codes then for UAG, which says stop. And then we have a missense, which is the T in the middle becomes a C, and we get a totally different amino acid. And then here we have the G instead of the T. Again, a totally different amino acid. And it is, this is the same type of amino acid, but not the same. This is a totally different type. We also have frameshift mutation. This occurs when nucleotides are inserted or deleted. It is worse when this happens at the beginning of a sequence. So here's a picture where this has happened. The frameshift, you can see we started here on the original DNA, and now we've shifted it to here, and this causes a whole abnormal strand of amino acids, so the correct protein doesn't work.